Good morning, Sterling Butler.
Hello, Mr. Sogo.
Good to see you here.
Excited.
Okay. We're at 11:06. One more minute and I will start this show. Okay, that's going over there. This is going over here. All right, it is 11:07. I will start this show. Co-host, please continue to monitor the waiting room, and as people arrive, please let them in. For those of you who are familiar with how these run, I'm going to present some information and then I'll pause at certain points to ask for questions. And the questions you ask, I'll try to answer them if I can. Please don't be shy about asking your questions. You can ask them via the text chat, or you can raise your hand. If you're familiar with Zoom, there's a raise hand function, and I can see who has a question, and I'll get to you, and you can ask your question out loud, which is great when you can do that. But the text chat will also work. I have to do a lot of things here. I am monitoring participants, so I'm going to relinquish that role to the co-hosts. I will try to stay on target here. This is not quite PowerPoint karaoke. If you're familiar with that, that's where the speaker doesn't know what the next slide is going to be. But I have not run through the entire presentation. I've just looked at bits and pieces, and I think it's good. But if I go oops, then I hope you'll forgive me. Here we go. The initial slide is a picture that I hope you're familiar with. This is an image of the virus. Structurally, it is like a ball. It's like a ball, but it has these spikes on the outside that are very important. We'll talk more about that in the coming slides. So this, you can see, is a cartoon of the virus structure that you saw in 3 dimensions on the previous slide. What's important is that it has proteins. So these that look like little artichokes are the spike protein. That's probably the most important one. It mediates the viral entry into human cells. So without the spikes, it cannot grab onto the human cell and it cannot enter. Then the next most important thing is this inside. The spheres are proteins, but they're surrounding the viral RNA. So the virus structure, the important parts are the proteins it has and the RNA, which allows it to make multiple copies of itself. The rest of this is an envelope membrane, and it is made of lipids with some proteins. You can see these little things here. That's the M protein. I don't know what it does. The E protein looks like a channel. It's got a pore through the middle, so it lets things in and out. Next slide is going to talk about RNA and DNA. This is essential to the subject I'm presenting today. So I hope this is review for you, that you remember your basic biology. That's actually a pun, huh? Because these are bases. These are bases. This DNA has this double-stranded structure. It's in every human cell. You have your DNA. The virus relies on RNA, which you can see is single-stranded. They made it into a helix in this cartoon, which is not realistic. It's not a helix, but the main point is it's single-stranded. Other than that, the structures are very similar. They're both using these 4 bases, which are usually abbreviated A, G, C, T, A, G, C, T. So here is an example of a DNA sequence. That's really what you care about, is not how many A's there are, but what's the sequence? What's the sequence? So this one happens to come from human insulin gene. If you were an ACR, you've seen this before. And you can see it's just a string of A's and C's and G's and T's. And it goes on and 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 on. The virus genome is about 30,000 letters, 30,000 bases. The human genome is 2 billion bases. So the virus is very small. It only has what it needs. It only has enough RNA to make what it needs, which is these 1, 2, 3, 4 proteins. And that's a very simple genome. It's actually more complicated than that. I think my next slide will show that it's more complicated than I made it sound. But that's important because the tests that we're going to be looking at today are focused on finding a specific sequence. Within here, if you said, let me read this line across from left to right, this sequence of letters probably only appears once in the human genome. Those of you who know statistics, it's 4 to however many bases as the exponent. So this is 4 to the 60th power, let's say. 4 to the 60th power is a huge number. So this particular sequence will only appear once in the human genome. And the tests for the virus need to find some sequence that will only appear in the virus, not in the 2 billion bases of the human genome. So it's finding a needle in a haystack, so to speak. So that's the challenge of the test. Find the virus gene. So this is where I was saying the real virus genome is significantly more complicated than I made it sound. These are the 4 proteins that I mentioned. The S is the spike, and the E and the M are membrane proteins, and the N is inside where the RNA is. But the actual genome has a lot more bases. But there's the 30,000 that I mentioned, 30,000 bases for the total RNA. And this is probably too complicated for you to understand. But the point is that nobody wants to read 30,000 A's, C's, G's, and T's. So they make what's called a map. And this is a map saying this particular stretch of the viral RNA makes the spike protein that's on the surface. The virus is made of RNA and proteins. The proteins are what you can see on the surface here. The RNA, you can't see it, it's inside, it's encapsulated. And this is, to understand everything I'm saying today, you need to understand these words, transcription and reverse transcription. It's not essential, but it helps. So DNA, the standard way human cells work is you have DNA, it gets turned into RNA, and the most important one for you to understand is messenger RNA. That's called transcription, and the messenger RNA is instructions to build a protein. So this particular protein on the virus, there was a gene inside the RNA that said make this spike protein, and so the translation turns the RNA into a protein. That's required to make a virus. It's required to make human cells as well. If all you had is DNA, you can't survive. I will mention reverse transcription, which is, as you can imagine, going backwards. You have RNA in the virus. You can turn it back into DNA using a special enzyme called reverse transcriptase. The virus doesn't have to do that, but the scientists do. The scientists who are studying the virus need to turn it into DNA because humans are very, very, very good at manipulating DNA and not as good at manipulating RNA. Boom. Okay, here's some more complicated pictures. But if we simplify and say what's important, the word polymer means put together many little pieces. So in chemistry this year, we talked about plastics are polymers. Do I have a plastic somewhere? Of course I do. Here's a plastic ball. It's a polymer. It's made of little tiny carbon-containing molecules that were linked together to make something that's solid. DNA is a polymer, and it's made of the individual A's, T's, G's, and C's. So here's a monomer, T, and it does have 3 phosphates, not very important, but it can be added to a growing chain. It can be added to a growing chain. T pairs with A, boom. The next one should be a G pairs with C, boom. A C pairs with G. So you make new DNA by extending the chain. And there is an enzyme that does that. It's called a polymerase. Anything that ends with ASE is an enzyme. Sterling Butler should know about that, huh? He used alkaline phosphatase. Oh, it's kind of fun to remember all your projects. So the DNA polymerase in humans copies your DNA, allows your cells to divide, allows you as children to grow. The test that I'm going to be discussing today is called a polymerase chain reaction. So if you understand, polymerase is just this enzyme that chugs along, making the chain, making the chain, making the chain, making the chain. This more complicated picture on the right is the virus polymerase. So the virus itself needs to bring its own copying machinery, which is up here in the genome. This is the 30,000 bases of genome of RNA. And you can see color-coded. These are the proteins 7, 8, 9, 10, and 12, and 14 are in this structure to make the polymerase that will copy the viral RNA, copy it, copy it, copy it, copy it, copy it. The virus needs to do that to replicate. But the test I'm going to talk about here is going to be using polymerase in a test tube. So it's not in a living thing, it's in a test tube. And that's the polymerase chain reaction. Oop. So here is a short-ish video. Before I start it, he's in Paris. So this is from CRI Paris, Centre de Recherche Interdisciplinaire. It's not international, it's interdisciplinary research. I'm hoping in the cartoon that's here, you can imagine this blob is the polymerase. These are chains of DNA that are going to be replicated. That's the polymerase chain reaction. Polymerase chain reaction. So here we go. Going through there. There we go. That's Paris.
Hi everybody, welcome back to Synthetic Biology 1. Today is a big day for us because we are doing PCR, the polymerase chain reaction. I cannot emphasize enough how important this technique is for people who work with DNA. You will use it all the time to isolate pieces of DNA, to modify pieces of DNA, or to prepare DNA for sequencing. The star of PCR is DNA polymerase. We use it to do what it does best, replicate DNA. But not just any DNA polymerase will do. Your average DNA polymerase, like most enzymes, will denature at high temperatures. At 50 or 60 degrees, the protein melts, unfolds, and turns into spaghetti. As we'll see today, we need something that can take the heat. So, back in the old-timey days, like the 1970s, some clever people started isolating DNA polymerase enzymes from microbes living in hot springs, which are very hot. These bugs still need to replicate DNA, and they've evolved very special thermostable enzymes that will still work even when we heat them to very high temperatures. These days, PCR is so popular that companies sell different DNA polymerase variants with different properties, faster replication, lower error rates, and so on. Next, we need something for our DNA polymerase to replicate. This is called the template DNA. It could be genomic DNA that you've extracted from an organism. It could be a plasmid that you purified in a mini prep. Anything at all. Importantly, we need very little DNA to kickstart the reaction.
Whoa, what happened? What happened? Shame on me. Okay. Let's do that again. Hold that up. So the template for this reaction is going to be the virus. And you can see the slide here is saying template DNA, and that's where you need reverse transcriptase to turn the viral RNA into DNA that humans are really good at dealing with. But that's the virus.
We need very little DNA to kickstart the reaction. Even literally one molecule can be enough. Next, we have to tell our DNA polymerase where to start. You see, the enzyme doesn't just bind to any old piece of DNA and start going. Instead, it can only add nucleotides to the 3' end of a growing DNA strand. It looks for places where the DNA double helix is partially formed, but with single-stranded DNA hanging off the side. We can create these points using primers, short pieces of single-stranded DNA that give the polymerase a place to start. A PCR reaction uses 2 primers, one on each side of the piece of DNA that we want to replicate. Finally, we need some free DNA base pairs to feed the polymerase. These are the nucleoside triphosphates, ATP, GTP, CTP, and TTP, that the enzyme uses to build the new DNA strand. That's 4 things: template DNA, primers, nucleoside triphosphates, and DNA polymerase. We mix them all together, put them in a thermocycler, and make the magic happen. A thermocycler is just a machine that can heat or cool our test tubes to a programmed series of temperatures. By cycling through a series of temperatures, we can get the DNA to replicate over and over again. First, we heat everything up to 95 C, near boiling. At this temperature, the DNA double helix melts and all of the DNA becomes single-stranded. This is why we needed that fancy thermostable DNA polymerase from the hot springs. Your average polymerase would be destroyed by these conditions. Next, we cool the reaction down to about 55 C. At this temperature, the primers can bind to the template DNA at the position where the sequence matches. We call this process annealing. Then we heat it up again to about 72 C. This is the preferred temperature of the polymerase. So it does its thing, taking the free base pairs and adding them to the growing DNA strand. The forward primer replicates the top strand, the reverse primer replicates the bottom strand, and pretty soon we've doubled our quantity of DNA. Now we just repeat the cycle. Heat to 95 and separate the DNA strands. Cool to 55 and bind the primers. Heat to 72 and replicate the DNA. And over and over and over. Each time we go through the cycle, we double the quantity of DNA. And now we just repeat the cycle. Heat to 95 and separate the DNA strands.
Okay, so I'm hoping that had meaning to you. You're going to have a chance to ask me questions in just a little bit. And I can come back to this one if the graphics help me. Next slide shows what does that look like in the lab. So all ACR students, this should look like a familiar daily task. Lots of pipetting. So the volumes you need are very small. So we use these micropipetters, just a few microliters of each thing, but there are a lot of things to put in. There's the buffer, there's the DNA, there are the primers, there's the nucleotide mix, and there's also the enzyme. So it's a lot of pipetting. And you can imagine this is kind of slow. So I sped up the video, but it's a lot of things to put in. Okay, so that's what it looks like in the lab. After you've got everything in the tube, then you put in the thermocycler, which I'll show you in just a wee bit. That will be actual footage from ACR. But I think my next slide is question time. So here's the Jeopardy song. I'll start looking at the chat, and I'll start looking for people who raise their hand with a question. So think about questions from at this point and let me know what they are. Uh-oh. Come on. Okay, here's the first question I have in the chat is from Luca Elganian. And the question is, why can't PCR be done with just RNA if the first step is separating the DNA with heat? So let me answer that question. The polymerase used in PCR is specifically copies DNA only. It cannot copy RNA. If we were using the virus polymerase, we could just start with the RNA because that's what it does. It makes RNA out of RNA. So that's where, since PCR has been really well established with that polymerase from the thermophilic bacterium, that's where we need this additional step with the virus RNA. So there's one step to start it off, which is reverse transcription. That's, again, a viral enzyme, but from a different virus. And it those are called retroviruses. So like HIV is a retrovirus. It starts as RNA, but it makes a copy in DNA. So there are enzymes that will do this. Hope that makes sense to you, Luca. It's, yeah, this DNA polymerase can't copy RNA. Okay, next question. Luke Johnson. If we are on track to the structure of the virus, can we find out chemically what makes certain people immune? That's an excellent question. I will be discussing that in some detail next time. I'm not going to introduce that into this virus talk, but yeah, exactly. The chemical nature of what makes immunity is a very important topic. Okay, looking for other questions. Ms. Perkins says, what prevents the DNA strand from reattaching when it is cooled to 55? All right, excellent question. Excellent question. So it's in here. I wish I had the still, huh? I wish I had the still. But let me see if I can get to it in here without messing myself up. Okay, so right about here.
To about 72.
All right, so you first separate the DNA.
Next, we cool the reaction down
now you cool
to
it
about
down.
55 C. At this temperature, the primers can bind to the template DNA.
Okay, so see, the primers are binding in the cartoon, but why doesn't this strand get back together with this strand? It can, but the way that the scientists make it so it is unlikely is by flooding this reaction with primer. So there's maybe 1,000 times more primer than there is DNA strands. So then it's just probability. The probability of the primer binding is higher than that these two strands get together. And the amazing thing is that once the primer binds, the polymerase is really fast to get on it and go zoop and extend the chain. You'll see it in my next video, how fast it is. I am always amazed. It's only a couple of seconds. Okay, I'm going to go forward. We'll have another chance for you to ask questions in a little while, so please don't be shy. Okay, so we did that one, lots of pipetting. And we did that one. And so the heart of PCR is the thermocycler. As the name implies, thermo is heat, and cycle means it just goes in a circle. It repeats itself over and over again. This is PCR and ACR. And this comes from Raul Campos's team. You'll see the actual machine. It's one we have at LBHS. That's why I could get this film. And on screen here, you'll be able to see the sort of the program that it's running, temperature data, and on the x-axis is time. And I'll talk about this little cartoon here. It's fun. So there's the machine. There's the machine. There's the machine. It's small. This comes from mini PCR, so it's small. Those are the little tiny test tubes that you have to put all the right things in. And you can see that's probably only about 100 microliters total, maybe even 50. That's the heating block. And it's hooked up to the computer, which controls the temperature inside there. So it starts hot. Okay, here we go, here we go, here we go. So this is the temperature. Like it's already gone through one heating, and then it cooled off, and it has this little plateau here at 72 degrees. And then it gets really hot again, and it cools off, and you can see it's going to cycle. But here, this part of the graphic tells you what's happening to the DNA. So this is a strand of DNA, and this is another strand of DNA. So as it gets hot, watch what happens to the DNA. You can see it's separating, and now I'll let you hear it. There's a fan inside that's cooling it off. Temperature's dropping, and those arrows, these arrows are the primers. So when the temperature's dropping, the primers are supposed to stick to the template DNA. And then in a little bit, you'll see it go zoop across, and that's the polymerase copying the DNA. Zoop, there it is, just copied the DNA. And over here, you can see the DNA copies is 8. And in just a few seconds, it's 16. Now we're going to, once again, heat up and separate, and there's the next cycle. But this is exponential growth. Every time it cycles, you double the DNA. And that's how PCR works. It just gives you an idea of what it looks like in real life on a small scale. I'll show you the big scale soon. So then after it's done, you have to know what did I make? Did it work? Is there a virus there? How do I know? So I'm hoping this looks familiar to some of you. Electrophoresis is the name of the method. We have these at LBHS, and they are for separating DNA based on size. So this first picture here is electrophoresis. That's from the company that makes it, Mini PCR. So that's professional quality. And this over here is Raul Campos's team. And there's their DNA. So you see it looks about the same. This on the left is what are called molecular weight standards. Here they're labeled. So these are our number of bases in the DNA. So this is 500, this is 6, 7, 8, 9, 1,000 bases in the DNA. This one here, if you understand genetics, these are, I'm almost certain, are from humans. And the question is, are you homozygous or heterozygous for this particular gene? And this person has only this type of DNA and is therefore homozygous. They called it little a. This person has only this type of DNA, is homozygous for big A. And this person clearly has both types of DNA and therefore is heterozygous, one big A, one little a. It's not important that you understand that, but if you do, then you learn something in bio, great. So this would tell you two things. One, did I amplify anything? Because Raul will tell you there were many times when it was blank gel, nothing visible, which meant that the polymerase was not copying the DNA and therefore didn't get anything. For the virus, that's actually a very meaningful result. If you're saying, I tried to copy the virus DNA and I didn't get anything, That's what a negative result is. That means you don't have the virus in your body. We could not detect it via PCR. On the other hand, if you do see the band show up, that's the right size for the viral DNA that you were trying to copy, then you say, yeah, you are infected. That's what we were trying to find with this test is, did we get something to amplify that we can pinpoint that's virus? That's not human, that's virus. So this, as I said, is old-fashioned way. And if we had to do virus testing at Laguna Beach High School, this is how we would do it. But we are not going to do virus testing at Laguna Beach High School. And there is a more modern, better way to get data faster. So that is called qPCR. And the Q is for quantitative. And this relies on fluorescence. So this is from ACR here, this picture. This was, I think this is Joey Ravenna's picture. David Wong also worked on this project, so he'll recognize what this is. This is fluorescence. These two tubes are under a UV light, and you can see this one glows. That's fluorescence. You've seen this in a lot of places, probably. Whenever you say, I used a black light and something glowed, that's fluorescence. The way the test works is that you look at fluorescence over a number of cycles. So this is the PCR thermocycler, 1, 2, 3, 4, 5, 6, 7, 8. Each time it cycles, it doubles the DNA, and you look for fluorescence in real time. So you get a graph, and if your graph looks like this blue one, that means that the PCR is amplifying DNA. And if you were doing a virus test, you would say, yep, you've got the virus because we're seeing the DNA that could only be viral DNA, can't be human DNA, is replicating in the PCR reaction. So I'll show you, my next slide has a video that'll explain where does this graph come from. And actually, I think maybe the next slide is this. Let's find out. It is. Okay, let's hope this works. So this is from Ravenna et al. Because to understand the next slide, you need to understand what is quenching. Let's see if this will work. Okay, it's going to work. You can see on screen a fluorescent sample, and ACR students will recognize this is a pipette. This is a pipette tip. It's a P200 pipette tip, and it has something in it that is a quencher. So things that are fluorescent, sometimes you can add a second chemical that will take away the fluorescence. It was bright, it will turn dark. So you'll see that here. That's the quencher that's been added. And as it mixes in, you can see it takes away the bright glow. So that's the phenomenon of fluorescence quenching. We've used it in ACR in a number of projects, mostly the peptide projects. So now you'll see that that's part of the qPCR technique. So this is from New England Biolabs. Raul is very familiar with New England Biolabs. And that's their little logo up there. Yep, let's give them credit. New England Biolabs. So you can see there's a graph similar to what I just showed you. This is cycles, which you can consider time. This is relative fluorescence. This is about 2 minutes. I hope it will explain things, and then you can ask me questions in a wee bit after.
In contrast to traditional PCR, where the presence or absence of a DNA sequence is analyzed upon reaction completion, quantitative PCR, or qPCR, monitors DNA amplification as the reaction progresses. In qPCR assays, reaction progression is monitored by an increase in fluorescent signal. The fluorophore can be a double-stranded DNA binding dye, most commonly cybergreen 1, or covalently attached to a target-specific probe, as in TACMAN assays. In cyber assays, the dye fluoresces when bound to double-stranded DNA, but displays weak fluorescence in the presence of single-stranded DNA. This results in an increased fluorescent signal as the reaction progresses, and double-stranded DNA is formed. The most common probe-based assay is a 5' nuclease assay, known as TACMAN. In TACMAN assays, the 5' flap endonuclease domain of Tac DNA polymerase cleaves a target-specific probe, separating a fluorophore on the 5' end from a quencher on the 3' end, resulting in an amplification-dependent increase in fluorescence. A thermocycler, coupled with a fluorimeter, modulates the reaction temperature during DNA amplification, while also monitoring the fluorescent signal at each PCR cycle, generating a real-time qPCR curve. There are 4 distinct phases within the amplification curve: lag, exponential, linear, and plateau. During the lag phase, amplification occurs, but the signal is not strong enough to be detected above background. The exponential phase is used for quantification because it produces a fluorescent signal measurable over background and contains an abundance of reaction components, permitting a detectable doubling event at every PCR cycle. When the fluorescent qPCR signal is detectable over the background fluorescence, a quantification cycle, or CQ value, can be determined. CQ values can be used to evaluate relative target abundance between 2 or more samples, or to calculate absolute target quantities in reference to an appropriate standard curve. qPCR can also be modified to detect and quantify RNA. Interpretation of qPCR data requires careful experimental design and analysis. For parameters on designing and reporting qPCR experiments, please read the MICE guidelines, which have been established to ensure consistent experimental design and data analysis appropriate for peer-reviewed publication.
All right, so that's the explanation of where does this curve come from. And my next slide is an attempt to explain that again. So if I think after this is a question time. So for this to work, you need a primer. The primer has to recognize viral RNA or DNA. I'm going to use those words interchangeably. It starts as RNA, turns into DNA very, very early in this process. So you need to know what primer to use. Here are examples of the sequences. I didn't count them yet. Let's count them. That's 3, 6, 9, 12, 15, 18, 22. So there are 22 bases here in this primer. That's enough bases that it will not randomly match a segment of human DNA. It will be unique for the virus. And so this is the other primer. It looks about the same size. And those are in my cartoon. There's a forward and reverse primer. Boom, boom. Then the idea of the fluorescent probe is also 3, 6, 9, 12, 15, 18, 21, 24, 26 bases. This is again going to be specific for the virus. And it sits here in the cartoon. TACMAN is probably a pun on Pac-Man. TAC is the Thermophilus aquatis. That's the species of the bacteria that makes the polymerase that works at really high temperatures. That's why it's TAC. So then this sits in the middle. It only binds to virus DNA. And as the polymerase starts chugging along, when it reaches the TACMAN probe, the polymerase itself starts chewing up that 26 base nucleotide. Yeah, I'll call it a peptide. Sorry, I'll just call it a nucleoside sequence, a DNA sequence. That's probably the best. So the polymerase, when it gets there, starts chewing it up. the C comes off first, the T comes off. It just cuts it as it's going along, saying, get out of my way. I'm trying to recopy the entire DNA, get out of my way. This graphic will look very familiar to those of you who worked on the peptide project. There's the sun and there's the cloud. And originally they are linked together at the two ends of this DNA primer sequence. And the one that glows, I highlighted in green and yellow, and that's the sun here. And the cloud is the black hole quencher. That's what BHQ stands for. That's Q in the cartoon. So it only gets separated if it was in the way of the polymerase, and it would only be in the way of the polymerase if the polymerase is copying the viral DNA. So this is a very clever system. I was so happy when I learned about it, because I thought, really, this is what we do in ACR. It's amazing. So there it is. The polymerase in the end has copied the entire viral strand here, and in the process has released the fluorescent molecule. Let's see what's next. I don't know what's next. Ooh, okay. That is scaling up. I think I should stop here and ask for questions. So once again, I'll monitor the text and I'll look for people who raise their hands. And please ask me a question about anything I've presented so far. I don't have a Jeopardy song this time. Shame on me. David has a question. Did I do it? I'm trying.
Can you hear me?
I hear you, David.
Yes. Hi. I have two questions actually. So on this graph, where did the TACMan probe came from? Like where did it come from?
Okay. It's on a slide that comes later, but the primers you order. So there is a company called Integrated DNA Technologies, IDT, and that's their whole job, is to make sequences of DNA for people who want to do this kind of work. So they can make this probably overnight. So you say, I need this tomorrow, and they'll say, we'll get it to you tomorrow. We used IDT to make Raoul's primers, and it was amazing. I was so surprised. It's like, here's your 20 nucleotide sequence. How much does it cost? $30. You can have it tomorrow. And I think they were in San Diego, so it's local. So there are companies that make DNA sequences, whatever you want, custom built, really fast. Thank you for the question, David.
So I guess what I meant was, like, how is it already on the DNA sequence? Like how?
Okay. Ooh, can I do that well? All right. In the beginning, can I take this away? No, I can't. Ooh, I can if I escape, huh? What will happen if I do? Let's find out. Okay, what will happen if I do? Let's find out. All right, so I added this polymerase here. It wasn't in the original graphic. Boom, it's gone. It doesn't exist. The TACMan can only bind if these two black strands of DNA are separated. So you have a whole bunch of TACMan probe just floating around. It's not bound to anything. Then at the high temperature, the DNA strands separate. And as it cools to 55 degrees C, the primers stick where they're supposed to stick, and the TACMan sticks where it can stick at that 55-degree temperature. So they're just floating in the reaction mixture, and they're dark because they've got the cloud attached to the sun. They've got the quencher attached, so they're dark. They're not fluorescing. And then as you cool it to 55, then they bind, and they happen to bind in the path of the polymerase. So as the polymerase goes through in those 5 seconds at 72 degrees, the polymerase chugs through and splits that one. And you have lots of these in reserve. So on the next cycle, you can do it again, 55 degrees, stick, chug, fluorescence appears. Does that answer your question, David?
Yeah. So I just want to make sure I understand it. So the primer is what finds the virus. So I guess they designed the primer so that it will match the sequence of the virus, but the TaqMan probe just attached to anything or everything.
No, it is also specific for the virus. Yeah,
I see. Okay.
so that's essential.
Okay.
That your primers are for the virus, because otherwise you start amplifying random human DNA.
Yeah.
It's useless.
Sure.
And you also need the TaqMan probe to be specific for the virus, because otherwise it won't bind in the right place to release the fluorescent signal. So this, let's see if my next slide, no, it isn't. So in order to do this, you need the viral sequence, and that did come from China in early January is when the viral sequence was reported. So people could start doing this science. And I'm amazed how fast it has progressed. And usually science is slow. And it takes a year to say, well, now we have a result. And here it's in less than 3 months, there's already a lot of results which are meaningful. Okay, can I see the chat? Let's see, chat. Okay, nothing in the chat that I can see. I'm going to lower your hand, David. Looks like I should go forward, and there'll be another chance for you to ask me questions in about 5 minutes, I hope. Okay, let's go forward. So the problem with PCR, as I called it, the old-fashioned ways, it's way too slow. It's too labor-intensive. One person pipetting for one sample is too slow. So the modern way this is done is with these replaced test tubes. So this is a 96 well plate, 8 by 12 is 96. You can say this is patient, you could say this is patient 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You get a lot of patients all at once in one 96 well plate. Usually though, you have to do controls. So those of you who have been working on the biofilm project know all about plates and controls. So let me show you next the more modern way to do this, which is with a robot. So I won't show the whole video here, but this is called high throughput qPCR. Biorad's a big biotech company. Instead of one person pipetting one at a time, the robot can do everything all at once. So you tell it what to do, and all the reagents are there in those little bottles. So all the enzymes, all the nucleotides, they're already there, and the robot can pipette much faster than any human. Probably the robot makes many fewer mistakes than a human would. And so this is how you can run, instead of one at a time, you can do hundreds at a time. So it has all the chemicals that are needed. It has the thermocycler that's needed. These are expensive machines, but you can see how it works. So it's much better than person one at a time. Okay, so that's probably enough for that. All right, I'll try to finish up fairly quickly here. I've done all the exciting part, but this is important too. So testing in the United States started very slowly, but now the big companies that have these machines are doing the testing. And so instead of maybe 100 tests a day. Now they're saying we can do 300 tests a week, 300,000 tests a week, 300,000 tests a week. And this started just about a month ago. Part of the reason for the delay was that the United States was not prepared for emergency testing. So usually the FDA, Federal Drug Administration, says, well, you need to prove that your tests are accurate, that they're going to be reliable. Before we can let you test, you need to show all that data. And that wasn't really the right response in this pandemic crisis. Needed to get testing done quickly. But this is from LabCorp, and I enjoyed reading this. It talks about the real-time reverse transcription PCR reaction. Here you can see they needed the reagents. The primers came from IDT, Integrated DNA Technologies. If you are an ACR, you know we get a lot of stuff from Fisher. And so there you go. You need your reagents to do it. They're using 3 different segments of viral RNA to look at. So that's what this N1, N2, those are different segments of the virus genome. Limits of detection are important. So how little virus is needed to get a signal, that's clearly an important number to know. So LabCorp did their test. And they did dilutions in 96 well plates. And this is the wrong number, but the idea that you make it more and more dilute, and you can see it starts dark blue, so this has got a lot of virus RNA in it or DNA in it. Probably RNA at this point is the right word to use. A lot of viral RNA here, less. And they did 2-fold dilutions. So then you say, how little is too little to get a signal? That's what they're calling the LOD, limit of detection. Their conclusion was, I'm going to average here, but it's about 10 viral RNAs per microliter. This is the nasopharyngeal swab. So this is the most commonly done method of collecting the sample, is you stick a Q-tip into the nose of the person, you pull it out, and it's covered with gook. And the LabCorp is saying, if there's 10 viruses in each microliter, we can detect that. If there's less than that, we might not detect it. So that's how you can get false negatives. Some people who clearly have the disease, but it didn't detect in the test, they might not have got enough virus particles in the swab. And then this is LabCorp data, which I found very encouraging because they're trying to prove their test works. So they didn't do real virus. Well, actually they did do real virus, but they didn't do real swabs from people who might have the virus. Instead, they said, we'll make a mock mixture of, I'll call it snot from a healthy person, and we'll put virus in so we know exactly how much we put in. And so they did 1x limit of detection, 2x limit of detection, 4 and 8. So these are 2-fold dilutions. And then they said, we didn't put any virus in this sample here. And they're saying that in their PCR testing, they got 100% accuracy on everything that should have been positive was positive, and everything that should have been negative, no virus here, was negative. So this was encouraging for me to see that in LabCorp's hands with the maybe a little bit cleaner sample than it would be in real life, because they put the virus in artificially, but their test worked flawlessly. All right, so that's the end of this show. I'm about out of time, but yeah, I will welcome questions, and if you want to stay on and ask me things, that's great. Next time, which will be Friday, I think, of next week, I get to talk about the viral proteins and human antibodies. This is the new wave of testing that's being rolled out. It has more benefits than just looking for the RNA. It may tell us who's immune to this virus, who has developed immunity, and that's clearly very important as we go to the future. once again, if you have questions, please raise your hand. I'll also look at the text. Question from Charlie. Okay, Charlie has a good question. Charlie Dickerson. As different strains of the virus emerge, can the DNA change significantly so that the primers in TACMan no longer work? The answer to that is yes, that can happen. It is unlikely because these primer and TACMan positions were chosen. There have been a lot of virus samples that have been sequenced, and they do have modifications and mutations, so they're not all the same, but the regions of high variability are the ones that you avoid when you design your primers. So usually there are certain portions of the viral genome that cannot be changed. If you change it, it doesn't work anymore. So those are called conserved regions. So it is possible for the mutations to make it so your primers don't work. And that's one of the reasons they do 3 different sections of DNA to amplify. So we're not just relying on one primer to work. We're saying if primer 1 doesn't work, how about primer 2 or primer 3, which are in different sections of the viral genome. Okay, I've got 2 hands, one from Sterling. I'm trying to unmute you, Sterling.
Yeah, hi.
Good.
Okay, so when the whole idea behind quarantining and social distancing began, say a month and a half or 2 ago, there was a sort of consensus that exposing the virus to excessive amounts of heat either kills it or maybe deters its effects. What exactly is the cause for that?
Well, the data, I mean, you can do the experiment in the test tube, and I haven't seen the data for that, but I think you're referring to the idea of as the weather changes, the virus may become less infective, and that has not yet been supported with evidence because the virus is too new to have the data. And we haven't had a summer with coronavirus yet. What's it going to look like when we do? The flu is very seasonal, and so flu in summer, you're not going to get flu in summer in this country. I've been watching India because India is a hot place. They don't have the ability to social distance. I read the number, there's 1 million people in 1 square mile in certain areas of India. The population is 1 million people in 1 square mile. So the idea of stay away from your neighbors is not possible in that neighborhood. So far the numbers for India are not alarming. They're going up, but it's not the apocalypse. So I'm still watching. And if India does not go apocalyptic, it may be because it is warm there. And that's my unsubstantiated hypothesis. But a hypothesis is a possible explanation. So I'm watching India and saying, if they don't go, then maybe you're right, that heat does inhibit the viral transmission.
Okay, thank you.
Thank you, Sterling. David, do you have a question?
Yeah, it's a question, also kind of like a comment. Have you heard of the CRISPR way of doing this?
I have not investigated CRISPR.
Yeah, so they came out with a test. Professor actually at Berkeley, they did a way of using CRISPR to do this, which is much faster. So I know PCR is quite slow sometimes, take a couple hours.
Yes.
Even with a machine, it probably still takes that long. So they're using this thing where you can do it in 40 minutes, they claim. And they also don't need a nose swab. Obviously claim they can do that. They should just publish a paper on Nature, I think. I haven't looked into it. I just heard about it because Berkeley's trying to make this big deal. But people are saying that they need higher concentration for that. I'm just wondering if you're familiar with CRISPR and why it's faster than PCR.
I can't answer that question. I haven't looked at it, but you've given me something to look up, so I will.
Cool.
Yeah, there was the idea of speed is important, for each test. Speed is important if you're saying, I want to know for this small sample right now, which is what you want if you went to the doctor and you said, tell me now instead of tell me 2 days from now, which in some cases is very important. For me, the more important thing is just to get more tests done, and with the high throughput PCR, those machines are well established. So it's not like we have to invent a new technology. People have been doing PCR for, 20 years with great success. But thank you, David. I'm glad to see that you're also following up on interesting things.
Thank you.
Okay, not seeing any new questions. This webinar has been recorded, so I'll put it on the YouTube channel. If you have friends who you think would enjoy seeing it, it will be there. Thank you all for attending, and hope to see you again in the future. Bye-bye for now.
Thank you, Mr. Sogo.
Thank you, Joey.
Thank you.